almost looks like I've got rabbit ears here because of the butterfly wings. Hi, hello, and welcome. A Microbehunter here, all over here. And I'd like to welcome you to another Saturday live stream. Well, actually, it's not a real live stream. The video that you're watching right now has been pre-recorded. So I recorded this a few days ago. Let me turn on the background here again. And still, you're able to use the live chat function, okay? And this way you're still able to communicate with each other, and you're able to post questions to each other. But I will not be able to answer them live like I usually do because, yeah, as I mentioned, I've pre-recorded this video here. Today I would like to put those butterflies here under my stereo microscope. They look quite nice. I did not make those, okay? So this was a present to me. I have some mixed feelings about actually putting butterflies, preparing them like this. But this was actually a present for me. I don't really trust those names here entirely. I don't know. So this could be actually something that might not necessarily be always correct. I don't know why, for example, they would call this black butterfly here the red breast, or red beast, or red breast. Okay, I don't know really, and and this one here should be the chocolate albatross. Really, I don't know. I have some doubts here. It doesn't matter. Nevertheless, they look quite interesting under the microscope, under the stereo microscope. And I also would like to show you this water sample here, which basically I just let me put the butterflies away, which I just collected recently. And this one is actually a very interesting one. It contains an algae called Hematococcus. It's quite a red. It's got a nice red color, and I would like to show this to you under the microscope. And I also have another water sample over here, okay, with some other algae. And I also found out that there are some other interesting microorganisms in here as well. So we're going to compare those two, okay. And that's basically what I would like to start off with. Again, if you want to ask questions, please feel free to do so. But in this case, you have to trust that somebody else in the community will be able to answer them. Maybe if I've got time, I'll be also watching this video together with you. But again, I'm not live. So if you're new to this format, or if you're new to this channel, what I like to do is I like to put things under the microscope together with you. It's a pretty improvised. Video stream usually what I do. So this means that what you're actually seeing, I'm just doing right now on here, and I generally do not do a lot of preparation. And sometimes the things that we discover here are going to be quite interesting, and sometimes maybe not. I'm going to put a small sample here on the microscope slide, but before I do that, actually, I would like to show you how I collected this. So I did prepare a little video. I made this video, as a matter of fact, just recently. So here, this is basically a pond that I visited, it's a beautiful swimming lake. Of course, here on the side, look what I found here. There was a little rock, and in this rock is some rainwater collected, and it had this brownish reddish discoloration. So this is already a sign that something interesting might be living in here. So I took a sample of this into this very tube that you see here that I've got, and I took it home and I put it at home under the microscope, and I found some interesting red algae, and I would like to show those to you today as well. They're called Hematococcus. I know that because I've got a book, an identification book, and I was able to identify it like that. Yeah, so now the video starts to loop again. It was actually quite a nice, warm, actually quite hot summer's day, so it was quite refreshing to go a little bit into the lake here. Yeah, so this is basically a very interesting place to look for a specimen. In the meantime, you can see that because I let the tube stand in my rack over here, you could see that the solid material started to collect on the bottom here. And so this will be the part that I will put under the microscope. Okay, so this clear water you will not be able to see a lot. And for this reason, I'm going to take a long pipette. I go all the way in, and then I will simply take up some of this solid material here, put it on the rest goes back, microscope slide. I'll put it again here on the rack. I recommend that you leave it open. so that essentially does not grow anaerobic. And it's also usually then if this happens, if it goes anaerobic, then usually it starts to smell bad after a couple of days. And then I'll put a cover glass on top. This is a fresh box. Ooh, okay, look, fresh box of cover glasses. And yeah, as I said, what I'm doing right now is actually non-prepared. The only difference is that it's pre-recorded. And look, here these are the cover glasses, and I always usually shine them, let some light. Oh, you can see it. Some light reflect. It's a bit dirty. So usually what I have to do is I take some tissue paper or some, over here I've got some microfiber cloth, give it a little wipe. And simply I dry wipe it, and usually this removes any grease or any fingerprints or any sometimes even some water spots that are on here, some calcium carbonate deposits. It's a little bit better, but not perfect. So I drop it on here, and yeah, I'll be going up with the magnification quite a bit, so it's really important that there is not too much water here, because the distance between the microscope objective and the cover glass will be quite small. So I simply remove any excess liquid using a little bit of tissue paper. Okay, and this should then do the trick. Okay, so there is a very thin layer of water here, and then you know what? Let's put it under the microscope, and I hope that we're able to see the same thing as yesterday. So this is another microscope view. Okay, let me adjust the camera, the stage camera, also a little bit so that you see. Okay, so yeah, and I'm going to put it in here right away. And this is what I see. I have to, of course, refocus everything a little bit. Okay. Yeah, the arrow over here, let's remove the arrow. And now we have to search a little bit. Okay. And there is a little bit of movement going on as well. And yeah, we are going to have a closer look at some of the things that we see here. Okay. It could be that those algae that I wanted to show you, they were quite abundant yesterday, but there are not so many to be visible right now. So let's go up here. You see, it always turns a little bit. Darker, and you see that there are a lot of these round structures here. Okay, now I'm gonna switch the arrow on again, and these are basically algae called Haematococcus. Hemato comes actually from blood, right? Essentially this is a pigment, and this pigment is related to the carotenoids. So basically it's chemically similar to the orange pigment in carrots. And this actually causes sometimes some ponds and some water bodies to actually appear to be blood red. And that is if there's this algal bloom of Haematococcus. Yeah. So, and here we see already something in the middle right now, which I would like to show you. Okay, again, I have to go up a little bit with the brightness, because this one here, what we see over here, is actually also one of those, but this is the motile or the mobile stage. So this basically means that those algae are able to move around. Okay, they have two tiny little flagella here in front, and when the conditions are not right, then they will become immotile and they will form those dark round structures. Yeah, and you can also see carefully, if you look very carefully, that there seems to be some kind of a gel layer around it, right? Yeah, so that's also very typical for those and difficult to see, but sometimes you're also able to see two tiny flagella here in front. So actually there were many more of them in this sample yesterday. I'm going to go up with the magnification further a little bit. Okay, you know what, I'm going to do it like this. Yes, this is a little bit better. Okay, and it's starting to, it has this interesting shape, but occasionally at the very tip, if you look, then you might be able to see at the tip, two small little flagella, two small extensions that are able to, with which it's able to move. Very difficult to see a little bit, yeah. So, and there were lots of those in the sample yesterday. So I kind of expected this a little bit because when doing a little bit of research, I was able to find out that if the conditions are not right, then they will actually form those round structures, right? But I think this the moving stage is of course more interesting. So what I'm going to do is I'm going to go down again with the magnification, and let's have a look at some. Maybe there are other places where I'm able to see those. This one, I don't know if this is one of them as well here. We have to zoom in again. But let's go all the way up, and here we go again, and let's have a look. We always have to look a little bit if there is this gel-like layer around it, because that could indeed be a case. And of course, the pigment, I don't know, this one here again looks a little bit different, okay? So what I think I'm going to do now is because yesterday's observation was really so much, there were so many more of those swimming around. I think what I'm going to do is I'm just going to show you now a video of what I recorded yesterday. So this is also something that we basically have to learn here is as soon as you take a sample from the natural environment and if you put it into a tube, then the conditions are going to change, of course. Oxygen conditions are different. Then also, light conditions are different. So this can, of course, mean that the composition of the sample changes. So nothing surprising here. But simply because it was so impressive yesterday, I'm going to now quickly show you what I recorded. Well, so this here is now the fresh sample right after I collected it. So obviously many moving cells here. A few stationary ones like the one on the left side has already become a little bit, how do you call this? A stationary? No, in a resting stage. That's the correct one, the correct term. So here again, the same image now using DIC imaging. The first one was actually regular bright field. Here we're adding now a little bit of this bluish background. But I think then the natural color of those cells is a little bit lost as well. But still, it looks okay. And this here is phase contrast. And I really like this one for several reasons. Because now you can actually see the gel-like surrounding a layer around the cells, and you can also see the flagella a little bit better. And I'm going to pause this now, hoping that we're able to see them a little bit better. Sometimes a little bit difficult to see again. Okay, but occasionally you see those tiny little string-like extensions on one side. These are actually flagella that are for movement. Here you can also see it a little bit. They are very difficult to see also over here in bright field, but in phase contrast, like here, you were able to see this much better, right? And we can also see that there is this layer, around the cell here as well. And on the inside, the reddish pigment is, again, that's the one that is related to the carotenoids, and it also has apparently commercial value. So apparently it's also used in cosmetics. So sometimes those cells are being grown, so to actually harvest those pigments, right? And again, over here on the side, we've got the resting stage here. So essentially, when the conditions are not favorable, they will form more of these, and we've seen in the other sample that many of the cells have already become those resting stages. In between here, those tiny dots that you see here, these are bacteria. So you see, it's quite a nice sample. I did a little bit of research and it was indeed confirmed that in many of these rocks where rainwater has collected and they dry out very quickly, and also in some bird baths, you can actually find many of those hematococci. Yeah, and quite happy that I was also able to find those, and so that I'm able to share this with you. What I would like to do, however, because I cannot quite believe, or do not really want to believe, that we're seeing that few of them here. That's a higher magnification, right? I'm going to actually try another sample, and I'm going to now try to pick the sample from a slightly different place from the tube. Yeah. So again, here this is using a high magnification 60 times objective. Yeah, using again DIC imaging. You see, it kind of goes in and out of focus all the time because the layer of water was a little bit too thick, so it kind of is also able to swim vertically, and this causes the cells to go a little bit in and out of focus. Yeah, but I think you get the point. Okay, and yeah, quite a nice specimen. So what I'll do is I'll try another one. Okay, so I'll take another microscope slide. Okay, I have to switch again back to the desk view here. Okay, the desk view. Here we go again. Okay, and I would like to have now. If you look here, you're actually able to see that there seems to be a little bit some layers. It's a little bit darker up here and less dark down here. So there has been some kind of, depending on the density of the sediment, it kind of settled differently. So what I'm going to try is I'm going to try to pick a sample from the bottom here as well. And I don't know if I'll be able to see more or less. But I'll go all the way down. It looks almost like sand in here. Okay, so let's give this here a try. Okay, the rest goes back. Okay, let's rinse this out again, and yeah, let's have a look here. Again, a cover glass. Yeah, again, if you collect samples from a natural environment, do not wait too long. Put it under the microscope immediately, because there is, as always, a natural progression of organisms. Okay, so you see it's fairly thick. Yeah, so there are some sand grains in here as well, causing some bubbles. I'm not so much worried about the bubbles really, but the thickness of the water layer will actually make it very difficult to focus. But let's have a look. Here we go. Oh yeah, that's the one from before. Let's take it away. Yeah, you see many of those round structures now. So let's put this one under the microscope now. There we expect more sand grains here, and it's way too bright. Yeah, there's not a huge difference here, right? Yeah, a little bit of movement every now and then. Yeah, but by far not as many as there used to be. Here again. This is actually also quite fascinating. I do not want to go much higher than 40 times. Okay. Do you see the structure? It always has this interesting, strange extension. Let's put this down here. Again, I was also able to see this before. It has this very irregular structure. Look, there is this long extension, yeah, on one side, which I was not able to see before, yeah, in the previous one. Yeah. So it would be kind of interesting maybe to continue observe. We don't have the time now, but maybe to continue to observe this, how it's actually able to change, how it will change over the next couple of minutes or hours. Yeah. Yeah. So it's actually also quite fascinating to see how those round cells, how different in shape and size especially they are. Yeah. But all of them share the common red pigment. Yeah. Here another one, look. Still, yeah. They also do not seem to swim quite as vigorously as the one that I just showed you. See, and that it goes out of focus. Here it is again. There's another one over here on this side. Okay. Yeah. And here's another one. But also this one here has this funny shape. Yeah, so that is basically, also these algae, and they are algae, is quite fascinating to see that normally we think that algae are actually green, but they have this reddish pigment. And essentially, sometimes when there are certain algal blooms in bodies of water, in ponds, for example, it can actually look like, the water can look very red. And um, it's almost looks like it's been discolored like almost like blood. Yeah. So that's now also a nice one here. See, it's going out of focus again. Yeah, see, that's also one of the reasons why you should always try to have the water layer between the cover glass quite thin. It limits the movement as well, of course. If there's too little water, you're actually going to crush it, and this is also not nice to see. But yeah, I think you get the idea. Oh, there's another one coming here from the bottom, right? Yeah. So this is a little bit the sample, which is now, and you can now see how, essentially we compare it again to the video from yesterday. I mean, this is of course a huge difference, right? A huge difference in appearance. Yeah, yeah, and again here. Yeah, really nice. I kind of like this. Okay, so I think I'm going to move on now to the next water sample, which are basically some algae, some green algae that are also pulled out, of the lake. And again, in order for before I start putting it under the microscope, I would like to show you again a video. Okay, so it's always quite interesting to see a little bit the context from where I took the specimens from. Yeah, so that's basically again the lake from before. There's plenty of stuff growing on the side here. Generally, the water was extremely clean. This was but I was able to find those, and it says, well, maybe there are not so many microorganisms in there, because usually if there is so much water movement, then very often any microorganisms are going to be washed out. Yeah? So I did not have very high expectations of a lot of water life in here, besides the algae themselves. But every time when there's a lot of movement, it could be that any cells, this is quite some stuff floating on the surface of the water. But generally, if there's movement, it's always going to be kind of rinsed out and washed out. And that's basically what I collected again. Yeah, so I again took those very same centrifugation tubes, and I'm going to, because the video starts to loop again, I'm going to show you, here is the tube. Okay, let me quickly focus again. Yeah, and you can see that, I don't know, just some oxygen bubbles forming. Well, I don't know. Yeah? In any case, there is a little bit of some tiny dots that you're able to see moving around here as well. Yeah, so this could be some water fleas, water crustaceans, whatever, right? They might be a bit difficult to catch now. I might try that. Yeah, so strictly speaking, I have to use a new pipette. Otherwise, there's going to be cross-contamination. But you know what? I'll be cleaning out the water sample in any case, so it doesn't really matter a lot. Yeah, I go in here and I did not catch it. Okay, let's go in here again, and I think I should have caught one of those water fleas, and as a matter of fact, I even see it. I'm able to see this tiny little thing. And now the question is now how am I going to do that? What I recommend is you just put everything on here. There's a tiny little dot here, right? And this means I'm going to remove a lot of the water again. Okay. Yeah, I'm going to remove. And in order to check what, it's not quite, So this tiny dot that you see here, I don't know what this is exactly. It's too small for me, so I'm going to put it now under the microscope directly without putting a cover glass on top. Okay, so let's see. Okay, so this means I have to again move over to the scope. Okay, I have to remove the other slide. And of course, now I have to kind of find it without actually, this is going to be a challenge now. I have to kind of find it without actually looking through the microscope. do it a little bit darker. So he's, ooh, how many of you know this long algae over here? Spirogyra. So let me find it. Where is this? Ah, okay. Here it is. Yeah, okay. Ostracod. Yeah, they're slowly becoming a little boring because I've got so many of them already. Yeah, it's an ostracod. It's a crustacean. Actually, I was hoping for something slightly more interesting. I mean, of course, they're kind of fun to look at. Yeah. But I've looked at them in the last couple of weeks already so often. Yeah. So that is basically one of those water crustaceans. Okay. And they like to feed on detritus, decomposing material, whatever. But if I basically, what I would like to do now is I would like to have a closer look at, now it's moving still, okay. I'd like to have a closer look at those algae here, okay, because they have this spiral-shaped chloroplast, okay. So what I have to do now is I have to put a cover glass on top, and what I'm going to do is, however, I'm going to, if I put a cover glass on top of it, I'm just going to crush it, and it's going to die, and because people don't like that if I do that, okay, for this reason I will simply take it up again. It's still here. I will take it up again. It's still here. I don't know if it's gone now or not. So now it's gone. Okay, now it's in here. I'll put it back in here. Okay, and now I can put a cover glass on top. So that we can really go up with the magnification again and have a look at some of those algae at close up. Okay, I'm just wiping the cover glass again. Yeah, and I think just from my personal view, this live stream or not quite live stream, yeah. I think, I don't know, tell me how you like it. Again, I'm not able to respond to your questions as I normally do, but at least I think maybe the general atmosphere of the live stream still is similar or the same. Huh? So I have to now have a look if I'm still able to find some of those algae here somewhere. So let me go down with the magnification. Let me remove the filter. There used to be so many. Now here it is. Here we go. Okay, here is at least one of them. Okay, so these are pretty huge, long long filaments. Okay, so this is the one that I would like to have a look at. And we go up with the magnification. We have to refocus. And I don't know if you just also watched my video on cyanobacteria, which is just recently published. Because the cyanobacteria are photosynthetic bacteria, and they were once misclassified as algae. And what you see here, that is actually a true algae. And they are not bacteria, but they're eukaryotes. Spirogyra, it's called. And let's go up yet further with the magnification. Oh, I love it. Look at this. Beautiful specimen here. Okay, and I'm going to center this again here. And let's have a look here. So where is this here? You see that this here is one cell. Okay, you can actually see that the ends of the cells here. Yeah, so that's one cell. And inside the cell, you have this spiral shaped. It's not. It looks like it's zigzag, but actually it's not. It's kind of it's spiraling. Okay, and when you focus through it, then you're going to see that this one here is in focus. And look what happens when I change the focus. Okay, when I change into which direction of the turn. In here, and now the other one is in focus, right? Because it's spiraling and top bottom. Yeah, and that is the chloroplast. Okay, so that's the chloroplast. There must also be the nucleus somewhere in there. I don't know if this is the nucleus over here or not. The nucleus, because it's not stained, it's transparent, therefore it's more difficult to see. And the chloroplast is green because it's filled with the chlorophyll with a green pigment for photosynthesis, of course. Okay, so I'm using again DIC imaging, and what I will do now is I'm going to now remove the DIC filter. I can go down a little bit also here and this is how it looks in bright field. So if you, most of you are probably going to see it like that. And here you probably can see also the natural colors a little bit better. Yeah. So DIC and phase contrast, they add fake colors, so to say. And this can actually distract a little bit. So this is again in regular bright field. And now I'm going to switch over to phase contrast simply to show you how this looks like. I have to open the condenser, and this is in phase contrast. Also looks kind of nice. Yeah. So yeah, 40 times phase contrast. Yeah. Maybe my head is a little too high here so that you're not able to see properly what's going on up here. Okay, so yeah, just wanted to show this to you. It's yeah, and generally what I always do, and I already mentioned this before in some of the previous videos, people keep on asking me, how come your images are all at high contrast and so good, and what camera are you using? And I'm telling you, it's not the camera. I mean, of course, the camera is also important, but that's not the main thing. The main thing is that you actually prepare the sample properly, and also, and that's what's not possible now, but in my videos where I also increase the contrast a little bit. So using this, color contrast, and this really adds a lot of quality. To the image. So that's basically just one of the algae in more detail. But I would like to now go back again to the sample, and maybe I'm able to see some other interesting organisms, because yesterday I don't know if I'm able to find it again. I found Paramecium buzeria, which is also quite nice to look at. I don't know if I'm able to find it again today, but let's give it a try. Okay, so let's go back to the desk view here, and let's add another water sample here, and I would like to now take some algae directly from here, and one way of doing that is simply by kind of going in and pulling out a sample. And putting the sample on here. That's of course way too much. Okay, I have to find some tweezers over here. And usually what I recommend doing is you take a little sample here. Okay, like this. Maybe you want to cut it off. And this one goes back. Okay, and I need a little bit of water as well. And let's try the following. Usually you have those so-called teasing needles, where I don't have those, because this one also works fine. All you do is you carefully separate this. Of course, you might be doing a little bit of damage to whatever is in here. So, but generally I think this way you can see the things actually much better, because otherwise there's so many algae in here, so many filaments, that it's difficult to actually see. And I don't know, this is a huge water droplet. I also have slightly larger cover glasses as well. Okay, they definitely do need a little bit of cleaning. So and let's try this here. It's also a fairly thick specimen. Carefully press it down a little bit. Let's give it a try. Okay, let's give it a try. Scope view. Okay, that's the one from before. Okay, I have to take this out. We put the other one in. Okay, wrong filter. Yeah, this here, this here, this here, this here. Oh, okay. Here we go. This looks already. Oh, look at this. Okay, you see. Oh, it seems to be carrying some eggs here on the back. Okay, you see there are plenty of living things here. Okay. Okay. So let's have a look. Okay. Lots of stuff. Usually you will find those, a lot of life next to the algae. The reason being that those algae are primary producers. So this actually means that they are producing, catching sunlight, doing photosynthesis, producing organic material. And this basically means it's the beginning of a food chain, and there is plenty of food present. Oh, here is another one. Okay, you see here it's a little too dark, everything. Yeah, that's why it's important to kind of always kind of separate it a little bit of what the specimen. Yeah. So I saw some movement here as well. Okay, what is this here? It seems to be a little worm. Looks a little bit like Iolosoma, which I've also found. It's a short one, however. Yeah. It's difficult to follow because it's kind of hiding among the algae. Let's do a quick scan of air bubble. Those bubbles probably will increase over time because the algae produce oxygen, and therefore the oxygen will collect and the bubbles will grow. This doesn't really disturb us. Yeah, and what time? Oh, I'm using now the 10 times objective. It could be that there is some. Yeah, I don't know what this is here. And let's keep on moving around the slide. I'm hoping to find the paramecium, but if not, then it's also fine. Yeah. So otherwise, let's go up a little bit with the magnification just for the fun of it. Okay, because this also looks kind of pretty. Yeah, we see a lot of diatoms. Okay, so if you I bumped into it. Those here, these are diatoms here. They have the silica shell on the outside. They look also quite nice, and you can see that they like to, at least in this case here, they like to attach to a surface. Yeah. So pretty big entanglement of algae and whatnot. But I think there is of course quite a bit of smaller ciliates and cells moving around. But I was actually hoping to find here also diatom, 2 of them here. And here in the back, you can also see a lot of background. All of these things here are diatom shells as well. Look, do you actually see that there's this movement? Okay, there seems to be something. What is this? It's again one of those guys here. Where is it? Gone. Here it is. It's a little out of focus. Here we go. Yeah. So, yeah, I think you kind of get the idea, I hope. Yeah, so let's go down a little bit again with the magnification, and that's for your for if you're new to this, for your info. That's basically what we hobby microscopists like to do. That's simply observing the things that we see here, And if there is nothing of interest here, ooh, this seems to be some kind of a colonial algae here. So let's go up with the magnification right in the center. Okay, yep. Looks a little bit similar to Pediastrum, which is the name also of an algae. Yep. Yeah. So that's yeah, an algal colony. Here the individual cells again. Yeah. And there are plenty of those also around. Yeah. So and that's basically what that's what I was just saying. What we hobby microscopists like to do is we just like to put samples under the water, under the microscope. Very often water samples because they're quite interesting to look at. Yeah, and then we just basically do a little bit of observation, and sometimes there are a few interesting things that we're able to find, and sometimes there are quite a few new things in there that were a little bit unexpected. And with the help of an identification book and with Google and Google image search and so on, we're able to kind of narrow down and identify some of those organisms that we see here. And in some cases, we are able to always see the same ones. For example, over here, I did not find, at least up to now, no rotifers. Yeah? Rotifers are micro animals. But in other water samples, the rotifers are very abundant. But again, this does not really surprise me that there is not so much movement going on here, because these algae, as I mentioned before, were kind of almost drifting a little bit in water. And if there's water movement going on, then the microorganisms are flushed out. Yeah, and I think this also explains why so many of the diatoms actually are connected to the algae and to other surfaces. Yeah, otherwise they would be actually flushed away. Yeah. So, yep, here again, lots of debris. Yesterday, I actually was. It's a question of luck. Of course, I think I was also able to find a flatworm. And so on. And if you now leave the water sample in here for a certain amount of time, then you're also going to see that the composition changes. This one over here, no, this is not. I thought this was the shell of an amoeba, but yeah, here nematode worms. They're always. What's this here? Was something? Ah, look at this. Ha! This looks like a flatworm. I'm happy to have found this. So have a look, okay? 2 eye spots here. This dark thing in the center, that's the digestive system, the stomach. Yeah, and yeah, it's now hunting for food. I'm happy that I found this. Okay, you know what? Maybe it paid off to be a little bit patient. Yeah. Let's follow this guy around a little bit. I've actually seen it. Some of them hunt and actually eat other microorganisms like Paramecia and so on, pretty aggressively. It was actually sometimes really gulping up the food and taking out a bite. Yeah. Let's follow the guy. So fast. Where is it? Ooh, I lost it. Okay, go down with the magnification. I'd like to see it. I'd like to find it again. I'd like to find it again. You see, they're so fast. Here it is. Here it is. Okay, it's a little out of focus. Okay, let's remove the arrow. Yeah. Let's remove the arrow. Yeah, those flatworms generally have a very high ability to regenerate. So if you were to cut them in half, it's not unusual for both of them to completely regenerate and form 2 separate individuals again. Yeah. So all of this is has been documented. And of course, they are also present in a variety of sizes. I think that this one is actually a rather small specimen. One of the fascinating things here that surprises also me as a biologist every now and then is that, of course, those flatworms, strictly speaking, are not microorganisms. They are true animals in the sense that they are multicellular. And now this basically means, here again the crustacean, this basically means that those flatworms are composed maybe of several thousand cells. Those cells must be quite small. Now compare the size of those cells now to the size of the cell of an algae, for example. Yeah. So you see that there is also, concerning cell size, a huge diversity. It didn't want to eat the worm. So what I would, now it's not possible because I have a cover glass on top, but what I probably would do now is, otherwise is I would probably take a small pipette and then try to suck it up and transfer it to a clean microscope slide without so many other things around. However, this might give a nicer image because there are not so many disturbing algae and other debris and cell fragments on there. But then the movement is also more unobstructed, and usually they're kind of moving around the microscope slide even faster. And then this might actually make it more difficult to chase it down. Yeah. But in any case, I was kind of happy that we've found this guy here. Yeah. Yeah. As promised, these type of streams, live streams, or pre-recorded live streams are deliberately a little bit improvised. Yeah, so I'm not planned. Yeah, so it's a pure coincidence that I was able to find this one here as well. Yeah. Nature is extremely diverse. Yeah, so here again those two. So this guy is kind of searching around, and I think what I'm going to do now is I'm just going to move on. I think we're just going to put the how do you call it, the butterflies under the microscope, doing a little bit of stereo microscopy as well. Yeah? And maybe last increase in magnification. Oh, you see, bad, lost it again. Yeah, it's somewhere. Yeah. Here. No, that's the other one. Okay. I'm going to basically switch over now to my stereo microscope, and I would like to put some of those butterflies that I got under the microscope. I have to go out and get them. Yeah, here they are. I have to also move all of the other equipment here out of the way. Yeah. And here it is, okay? So what I would like to do is I would like to show you some of the scales of the butterfly because they have also a very characteristic shape. And over here I have, on this slide here, I've got the wing of a mosquito which I mounted some time ago. And I would like to also compare the scales of a butterfly with the scales of a mosquito. Okay, because you're going to see that there's an interesting similarity. So we're going to go into the stereo view, and here we go. I'm going to put it beneath the stereo microscope. Yeah, and let's always start at the lowest magnification here. Let's focus a little bit. Yeah, the image quality might not be the best because there's a glass surface here as well. Okay, but it's gonna be enough for essentially what I want to show you. Here's the head. Yeah, here's another one. Okay. Yeah, and here you see that there has been actually some deposit on the glass on the inside. But I found that this one over here actually is a very good specimen because of the high contrast between the dark and the white scales here. Okay, so let's move up. Let's go up with the magnification here. Okay. Yeah, I did not properly adjust the microscope. Normally it should stay in focus as you increase the magnification. And here we already start to see some of the scales. Again, it's a little out of focus. Here we go. Okay. So that's the maximum that I can have here. Okay. So the white scales, white on white is very difficult to see, if not impossible to see, but at least you're able to see the dark scales on the white background. Okay, and if you put dust samples, for example, under the microscope, or if occasionally even in water samples, we're able to see those scales. And then every time when you see those, you actually know that these must be the scales of a butterfly or of another insect. Yeah. So they are quite common, butterflies, moths, and so on, but not only. And those scales are relevant for a variety of reasons. Of course, they influence the aerodynamics of the insect, but also something that I read, interesting, when a butterfly like this is caught in a spider's web, then essentially the butterfly has an easier time to escape because some of the scales will actually stay on the spider's web. They will kind of break off and the butterfly is able to escape. So, yeah. Here again, quite nice. Yeah. And here we're also able to see individual scales, and you see that they're not arranged and this means that they actually have broken off, separated, and that's why they're scattered around here. Okay. So I quickly also wanted to show you one from another butterfly, maybe the one over here. I think we always start best again at a low magnification here. That's the head. And when we go down, and here we're able to see again the scales. Okay. And over here, the last one. So I think you get the idea. Butterflies are quite nice to look at. Again, I would say the image quality is not quite very good in contrast because of the glass that I have here. Okay, covering the whole thing. But I would like to now show you the slide of the mosquito wing that I recently made. And for that, you know what? I can actually show it to you also here first. Not easily visible yet. Okay, and then I'll put it into the compound microscope as well. Yeah. So these are basically the butterfly, not the butterfly, the mosquito wings under the stereo microscope, and you might already be able to see some of the tiny scales here. Yeah. Some of them also have broken off. And now let's put this under my compound microscope because we're able to see this much better. Okay. So compound microscope is here. Okay, it's still the one from before. No, it's stuck. It's dragging along. Yeah. Some material. Another thing before I actually show you, do you see those things here? Yeah, these are bubbles, and sometimes they will start to form, seemingly in the middle of nowhere, but actually that's then because the cells, the algae that you have over there, they might actually be producing oxygen, and that's why that bubble might actually grow over the next couple of minutes. Yeah, we don't have time to. Here it is again. What a coincidence. Okay, so I'll show you the mosquito wings. Okay, so I just added the slide here, and we just move over directly. Lots of dust on here. I have to find it again. Here it is, at least one of the two. Condenser goes in. Magnification goes up. We have to focus. And here we see the scales much better. And yeah, if you think that they kind of look similar to a butterfly, to the scales of a butterfly, then you are correct. Because, and now I have to confront you with a biological term, those scales, according to my research as well, are homologous to the scales of a butterfly wing. It means they have the same origin. Yeah. So there is, And this means that if you, again, if you're able to see some of those scales here, some of them are broken off in a water sample or dust sample, then you know that these are from insects. So this here is the website inaturalist.org, and I think it's a great place also for microscopists to become active. There are already many microorganisms in the database, but not only. And it's also quite useful for helping identify microorganisms. I'm going to just show you the few observations that I've already made. I've not been very actively involved in here, but you can see that essentially already uploaded a few of these microorganisms here. So for example, here this is a tardigrade, a diatom. And what I would like to do is I would like to show you now how to upload also something. So what you do is you click the upload button on the top right corner, and you choose files, and then over here we've got some, and this here is a rotifer. I'm just going to upload this rotifer over here. And you click open, and basically, here it is. And what you do now is you don't know exactly what it is. So look, it already found it, rotifera. Okay, I have to include a date, so I'm going to include a date. I have to include a location. I'm just going to pick any location right now, simply for demonstration purposes, right in the middle of the ocean. And I can add some notes here, and I'm going to submit it now, right? And then it's in the database. Okay, so this is something that I kind of highly recommend, especially for those of you who are doing microscopy, and if you want to have a project a little bit to keep you motivated, then I really consider this a very nice website. Okay, of course you can also do searches. So if you go up here at the top, and if you type, for example, paramecium. At the top, then it's going to find a whole range of different paramecia. So let's click View Observations, and then it's going to list all of those uploads and images that are tagged with paramecium. So this is actually a very nice website, also to help you identify certain microorganisms. If you of course click on here, then you're going to get more information about the specimen here. Okay, so this is actually quite nice because especially if you kind of are looking for projects or for some community to share your images with, then I think this is a very nice place to visit. I have not been very active in iNaturalist, I have to admit. I spend most of the time that I have making YouTube videos, but I think it might actually be a good idea to become a little bit more active as well. Okay, so I'm going to leave it at that. I hope that this pre-recorded live stream was still quite interesting for you. I do, of course, would like to invite you to subscribe to the channel if you like these type of live streams and videos. Please do check out also the other videos that I've made in the channel. But for today, I'm going to leave it at that. I wish you all the best. Happy microbe hunting as always, and hope to see you again in the future in the next live stream. Bye-bye.